mechanism. So primary hemostatic mechanism, as we tried to summarize, state earlier in the last lecture when we were discussing about the role of the endothelial cell in blood clotting. We were talking about some aspect of it that are connected with primary hemostasis. So we're going to look at the steps that are involved in primary hemostasis. First, we have the transient constriction of damaged vessel mediated by reflex, neural stimulation, and endothelial release from the endothelial cells. So at the point where there is a cut in the body, what happens is that the blood vessels try to constrict, and that reflex action helps to narrow the vessel lumen to limit the amount of blood that is oozing out from the vessels. Now, the second step is involved platelet adhesion to the surface of the disrupted vessels. The von Willebrand factor will bind the exposed subendothelial collagen, and the von Willebrand factor is derived from the Weber pallid bodies of endothelial cells and the alpha granules of platelets. Then platelets binds von Willebrand factor using the glycoprotein 1B receptor. The next step, we'll still see this depicted with a diagram. The next step was the platelet degranulation. Degranulation, because of the adhesion of platelets to the surface of disrupted vessels, it causes shape change in platelets, okay, and results in degranulation, that is release of multiple mediators from the platelet. So the process of the platelet adhering to the ruptured vessels makes it to change shape, and this change in shape causes it to be activated. The activated platelets will then release some mediators that will help in the blood clotting. Some of them from ADP is release ADP from the dense granules of the platelet. And what this ADP does is to promote the exposure of a secondary another receptor, the glycoprotein 2B3A receptor on platelets will be exposed. Then it's also released thromboxane A2. Thromboxane A2, what it does is to promote platelets aggregation. Then step 4, platelets aggregation. Platelet aggregation at the site of injury using glycoprotein 2B3A, using the fibrinogen as a linking molecule. This results in the formation of a platelet log. So as you can see here, this is a summary and overview of the coagulation mechanism. Okay, we said that the coagulation mechanisms broadly divided into the primary and secondary hemostatic mechanisms. We also have the fibrinolysis as part of the overall coagulation mechanism. So we have the response to injury, first vasoconstriction, which is part of the primary hemostatic mechanism. So primary hemostatic mechanism, we have the platelet adhesion, platelet aggregation, before we go down to secondary hemostatic mechanism, where the plug that was formed in the primary hemostatic mechanism will be stabilized during this secondary phase of coagulation. After that, there will be fibrinolysis to dissolve to form the plug. The fibrin that is formed should be dissolved to return and maintain the vessel patency. So as I was trying to let us know earlier, this is a diagram trying to describe the primary coagulation mechanism, primary process of hemostasis. So here you can see the injured endothelial cells. You can see the injured blood vessels, injured endothelial cells exposing the subendothelial structures, and also the endothelial cell releasing some of its contents and exposing the subendothelial structure where we have the collagen factor, collagen. Okay. So upon exposure of these subendothelial cells, either one or any one of these can happen. Von Willebrand factor, which is exposed when the endothelium release, when the subendothelia is being exposed, will bind to the platelets. Okay, platelets will bind to von Willebrand factor using its glycoprotein 1b receptor. That can happen. Or the collagen also exposed in the subendothelial structure as the endothelial cells is being ruptured. The platelet can also bind the collagen using its glycoprotein 1a receptor. So either of these two can happen and will result in platelet adhering to injured blood vessels. All of them will lead to platelet adhering to the injured blood vessels. And once that happens, platelets will have a shape change. A shape change will occur, and that change in shape of platelets will trigger the activation of the platelets also. So when the platelets are activated, say they release some of their mediators, the ADP and the thromboxane A2. Well, we'll say that the ADP will cause the platelet to also express small receptors, glycoprotein 2B3A, okay, cause platelet to express these receptors, and these receptors to bind fibrinogen with these receptors. So fibrinogen will serve as a linking molecule, linking each platelet to the other. Okay, so platelets will be linked together, okay, leading to the aggregation of platelets. They used this fibrinogen as a linking molecule, and this was possible because of the activation of platelets and release of ADP, which causes the platelet to express glycoprotein 2B3A, which binds to the fibrinogen, and the platelets links themselves together using the fibrinogen as a linking molecule, resulting in platelet aggregation. This is platelet adhesion, platelet aggregation. So activated platelet localize coagulation to the site of injury. So this injury are localized at the site. This aggregation are localized at the site where the injury occurs. So, and that brings to end the mechanism of primary hemostasis, from vasoconstriction to platelet adhesion to platelet aggregation. So we'll look at the secondary coagulation mechanism. Okay, sometimes when you observe the secondary coagulation mechanisms, sometimes it may feel as if it is something that you cannot comprehend and is just unnecessarily complex. But not so, because as the various systems and pathways are being developed, it was discovered to be an elegant series of serine proteases that amplify a small stimulus into effective defense. So coagulation is controlled by a series of mechanisms, and its products are ultimately resolved in seconds in a well-contained metabolic process called fibrinolysis. So the process of secondary hemostasis or coagulation seems to be complex, but it has been discovered that it's not as complex and it's not as complex as thought. Okay, that the final result of this reaction is to convert fibrinogen that was used as a linking molecule in the primary hemostasis to insoluble strands of fibrin. Together with platelets, the fibrins transform a stable blood clot. Okay, so the secondary coagulation mechanisms are also broadly described, can be described by two pathways. Initially, we used to describe it using the intrinsic and the extrinsic pathway, or they call the waterfall cascade, or it can be described using the cell-based model of coagulation. Either of these two can be used to describe the secondary hemostatic coagulation. So the extrinsic and the intrinsic pathway, waterfall cascade model of blood coagulation. So it's this in this coagulation pathway, also called, some also describe it as the plasma-based in vitro coagulation. Some also described it as that. Okay. In the past, the two coagulation pathways were described, both of which activated factor 10 at the start of the common pathway, leading to thrombin generation, both of them merging at the common pathway. So the pathway were characterized as cascades. They were characterized as cascade in that one enzyme becomes activated. As one enzyme becomes activated, in turn, it will activate the next enzyme. That is the view of the cascade. Okay, so and most coagulation experts, they identified factor 12 as the primary step in coagulation, because this factor could be found in the blood, whereas the tissue factor forming the extrinsic pathway could not be found in the blood. And because of that, the reaction system from factor 12, okay, the reaction system from factor 12 that leads to formation of fibrin was described as intrinsic, okay, while that of the tissue factor pathway were described as extrinsic, because the tissue factor will not be found in the blood, while the factor 12 will be found in the blood. Because tissue factor is not present in blood, the tissue factor pathway has been described as extrinsic pathways. Okay, and as you can see from the diagram, the two pathways have the common pathway. Okay, so but this description of extrinsic, intrinsic, and common pathway are used extensively to interpret in vitro laboratory testing and to identify factor deficiencies. Okay. However, they do not adequately describe the complex interdependent reaction that occur in vivo. So the extrinsic and intrinsic coagulation pathway were seen as independent pathway, but later it was observed that it was not so, as we'll see when we describe the cell-based model of blood coagulation, so that the two pathways are not distinct from each other. As we can see, the tissue factor here activating factor 9, that is an intrinsic pathway. Okay, so the extrinsic and the intrinsic pathway used to describe blood coagulation, starting from factor 12, which is used to describe the intrinsic pathway because factor 12 is found in the blood, and the tissue factor pathway, the extrinsic pathway, because the tissue factor is not present in the blood. Both systems culminate in the common pathway that generate thrombin to convert fibrinogen to fibrin. Then factor 13 cross-links the fibrin monomer into stronger fibrin molecules. So the cell-based model of blood coagulation, we say that for years, the coagulation cascade was conceptualized as having two distinct points of initiation, labeled the extrinsic and the intrinsic pathway. Over time, however, it has become clear that these pathways does not function in the body as parallel, independent system. The observation that the tissue factor 7 complex activates factors in both systems. That is, it activates both factors. It activates factors in the intrinsic pathway and it activates factors in the extrinsic pathway. As we can see that the tissue factor 7 was activating factor 9, suggests that they are linked. This discovery, combined with an evolving understanding of the role of different cells, in particular platelets, has led to a cell-based model of blood coagulation. So as it was discovered exactly that the coagulation mechanism are not independent as was previously assumed, a factor 7, tissue factor 7, activating a factor in the intrinsic pathway, and also the role of platelets in the blood coagulation led to the evolution or description of the coagulation cascade using this cell-based model of blood coagulation. So the cell-based model of blood coagulation identifies the membrane of the tissue-bearing cells. It identified the membrane of the tissue-bearing cells and platelets as the site where activation of specific coagulation factors occurs. So it's now trying to identify the membrane of cells, tissue-bearing cells. Okay, so this model suggests a three-phase process. A three-phase process, unlike the extrinsic, intrinsic, and common pathway. It describes it in three processes: initiation, propagation, and clot formation. Okay. So the initiation occurs on tissue factor expressing cells. Okay, and it produces 3 to 5% of the total thrombin generated. So you can see the membrane-bound tissue factor activates factor 7 to activated factor 7, leading to the formation, because these tissue factors are born on the membrane tissue factor bearing cells. Okay, you can see the tissue factor here. Okay, you can see the tissue factor. It's membrane bound, so it activates factor 7 to activated factor 7. The membrane-bound tissue factor 7 complex activates both factor 10, you can see it here, and also activates factor 9. You can see it here, activating factor 10 and activating factor 9. You can see the tissue factor, membrane-bound tissue factor activating factor 7, leading to activated factor 7, forming the tissue factor 7 complex, okay, which now activates both factor 10 in the common pathway and factor 9 in the intrinsic pathway. Factor 10 converts small amounts of prothrombin, okay, small amounts of prothrombin, activated factor 10, activate factor 10 to become activated factor 10. Then activated factor 10 will now convert small amounts of prothrombin, factor 2, to activated thrombin, which then activates factor 5 and also factor 8. This is what happens in the initiation phase. Okay. So the next phase is the propagation phase. The propagation phase occurs on platelets, and it produces 95% more of the total thrombin. Okay, it produces 95% more of the total thrombin. Okay, and clot formation. So as you can see in this diagram, at this stage, greater percentage of the thrombin that is being formed in coagulation process are being generated. Okay, the central role of activated factor 10. Okay, on the surface of the activated platelet, factor 11 activates free factor 9, are still activation of factor 9, which then form the tenase complex with factor 8. This activated factor 9 and factor 8 activates factor 10, which in turn forms the prothrombinase complex with factor 5, phospholipid and calcium. Okay, with factor 5, phospholipid and calcium. This is the tenase complex. This is the prothrombinase complex. Okay, the thrombin that is being generated. Okay, okay, okay. So this is the tenase complex, as you can see in the diagram. This is the prothrombinase complex. Okay, so which converts a large amount of prothrombin to thrombin. You can see it taking a large amount of molecules of thrombin to form prothrombin to form thrombin. Each molecule of activated factor 10 generates approximately 1,000 molecules of thrombin, which we refer to as the thrombin bus. Okay, and this thrombin bus goes back to activate more factor 9. Thrombin amplifies the coagulation cascade through the activation of factor 9. So in this propagation phase, there is a generation of large amounts of thrombin that will go back into the coagulation mechanism to cause more formation of thrombus formation. So we have also the clot formation stage. The clot formation stage of blood coagulation. Okay, so here thrombin converts fibrinogen, all the thrombin that are being generated from the propagation phase will now convert the fibrinogen that was used as a linking molecule in the primary hemostasis to form fibrin, first of all, fibrin monomers. Okay, you can see the thrombin here converting the fibrinogen to fibrin monomers, also activating more factor 13. Okay, more factor 13 that will cross-link these fibers. So thrombin converts fibrinogen to fibrin monomers, which form fibrin polymers. The same thrombin also activates factor 13. The same thrombin, okay, converting fibrinogen to fibrin monomer, activating factor 13. The same thrombin, okay, thrombin activates factor 13 to activated factor 13, which cross-links the fibrin strand, as you can see in the diagram here, leading to fibrin scaffolding that, along with blood cells, forms a stable clot. So some blood cells, white cells, are trapped in here, forming a more stable block. Okay, because the platelet block that was formed in the primary hemostasis using fibrinogen as a linking molecule are unstable. They can be dislodged as the blood flows into circulation. They can be dislodged. They are not stable. So they have to be stabilized in the process of secondary hemostasis that converts the fibrinogen to fibrin monomers and then cross-linked by factor 13. Thrombin further promotes clot formation, although through activation of platelets. You can see the various functions of thrombin in the process of fibrin formation. It is converting fibrinogen to fibrin monomers. It is activating factor 13 that will cross-link the monomers, and it's at the same time activating more platelets to play a role in further coagulation. So this coagulation process are not without check. The coagulation process are not without check. The process does not go on indefinitely. So the process has to be controlled. Immediately the body achieved clot formation and there is a block in blood loss. The blood loss has been arrested. Okay, the process of blood coagulation has to be regulated. Regulation of blood coagulation. Okay, sorry. So we have the activated protein C pathway. This is a pathway of blood coagulation. Okay, protein C. Protein C is produced by hepatocytes and they circulate in the body as inactive in inactive forms in the plasma, and they are cleaved by thrombin. Okay, when thrombin is bound to thrombomodulin, a constitutive protein of endothelial cell membrane. Okay, protein C is also produced by hepatocytes. Okay, and 40% circulate as free protein S and 60% as bound and inactivated by the complement system. Okay, okay, but free protein S binds and stabilizes activated protein C, which is now bound to the endothelial protein C receptor. So the protein C in the body, activated protein C pathway, that is, we have this protein in the body, and the activity of this protein, the activated protein C is also digest and inactivates factor 8 and factor 5. Okay, it inactivates factor 8 and factor 5 so that they are no more available to contribute to the process of blood coagulation. So it's a natural process of regulating blood coagulation at the end of the stoppage of once clotting has occurred, the coagulation mechanism has to be inhibited, sort of, has to be inhibited. And one of the pathways through which that can be achieved is through the activity of the protein C receptor. Endothelial protein C is through the EPCR and APCR pathway. The endothelial cell membrane protein C receptor 1. Okay, and the activated protein C. Okay, this protein activates protein C, combining with protein X. The activated protein C protein S complex will inactivate factor 5 and factor 8. Okay, reducing the formation of blood clots. So as you can see from the diagram, thrombomodulin, okay, activating the protein C endothelial cell C proteins, endothelial protein C receptor, okay, which activates protein C, which we call the activated protein C, which combines with protein S to form the activated protein C protein S complex. This complex is now what's short of factor 5 and factor 8 from participating in the blood coagulation pathway. So we also have the antithrombin pathway of blood regulation. The antithrombin goes to regulate blood coagulation by canceling the activity of factor 9 and factor 10 and thrombin. As the name is, antithrombin. It goes to stop thrombin and factor 10 and factor 9 from participating in the coagulation mechanism. We also have the tissue factor pathway inhibitor. The tissue factor pathway, also from the name, it inhibits the tissue factor pathway and the protein S. factor 10 combination with protein S, it shuts off factor 7 and factor 10 from participating in the coagulation mechanism. So they are called the natural inhibitors, the antithrombin, the APC, okay, and the tissue factor pathway inhibitor. So we are going to also briefly look at fibrinolysis. We said earlier that as the blood clots are being formed, okay, there is also the need to also remove the plugs, the fibrins that are also being formed. It does not go on indefinitely. Blood clot does not go on. Formation of fibrin should not be allowed to go on indefinitely. It has to be controlled. Otherwise, it will block the process of blood flow. So at a certain stage of the blood coagulation, during the process by which the fibrins are being formed, as the fibrins are being formed, they act as stimulus to trigger off fibrinolysis. So as we recall from the secondary hemostasis, a series of protease reaction, fibrin formation is a series of protease reaction causes the conversion of soluble protein fibrinogen to insoluble fibrin strands by thrombin, leading to thrombus formation. The thrombin also activates factor 13, which stabilizes the thrombus by cross-linking fibrin. By cross-linking fibrin. The resulting fibrin mesh traps and holds cellular components of the thrombus. So some cellular components, platelets, red cells are also trapped there to form a more stable mesh. Now, fibrinolysis is the process that dissolves fibrin, leading to dissolution of the thrombus. Plasminogen, the precursor of plasmin, breaks up fibrin in the thrombus. During initial thrombus formation, plasminogen activators are inhibited during the initial process. Plasminogen activators are inhibited. Endothelial cells begin to secrete tissue plasminogen activator to start dissolving the thrombus as the structural integrity of the blood vessel wall is restored. Fibrinolysis must occur for normal blood flow to be re-established. See it. Drugs that convert plasminogen to plasmin are used to treat acute life-threatening thrombotic disorders such as myocardial infarction and ischemic stroke. So when you look at this diagram that tends to depict the process of fibrinolysis, we have the plasminogen activator, which converts plasminogen in the inactive form as an inactive factor, converting it to plasmin. And this plasmin has the capacity to break down this fibrin that was formed during the process of clot formation. Break it down. Now we have these D-dimers. Okay, so we use also these dimers in laboratory diagnostics. When you evaluate the D-dimer level of a patient, you're actually evaluating the process of clot formation. Maybe some people that are having thrombosis, that a myocardial infarction and stroke patient, well, because the clot has to be completed before the degradation product can give rise to D-dimers. So it shows that there is complete formation of blood clots, and we use it as a value in the laboratory to evaluate patients with clotting disorders. So fibrin formation drives fibrinolysis. So as fibrin are being formed, I say it acts as a stimulus. The fibrin that are being formed acts as a stimulus to also initiate fibrinolysis. So initially, all these zymogens, okay, are present in the plasma, the plasminogen, the tissue plasminogen activator. They are all there, but normally no interaction between them. But as the process of fibrin clots continues to form, as fibrin clots continues to form, the tissue plasminogen activator attaches to the fibrin clot to convert the fibrinogen to plasmin. So the plasmin that is now generated will lead to breakdown of the fibrin clots. Fibrin degradation products are being formed. So for fibrinolysis, it is the last stage of coagulation, which is the dissolution and localization of the last stage of coagulation is fibrinolysis, which is the dissolution and localization of fibrin clots. The blood fibrinolytic system comprises an inactive proenzyme, plasminogen, that is converted to plasmin. Plasmin degrades fibrin into soluble fibrin degradation products by two physiological plasminogen activators, the tissue plasminogen activator and the urokinase type plasminogen activator. The tissue plasminogen mediated plasminogen activation is mainly involved in the dissolution of fibrin in the circulation. Okay, the urokinase tissue plasminogen activator binds to a specific cellular receptor, resulting in enhanced activation of cell-bound plasminogen. So the fibrinolysis process are also regulated. The clotting process does not go on indefinitely. Fibrinolysis tends to regulate it. Then the fibrinolysis process also not allowed to go on indefinitely. There are also a regulation of the fibrinolysis process. So the coagulation cascade is a complex feedback mechanisms. Okay, so fibrinolysis are also regulated. The inhibition of the plasminogen activator are inhibited by plasminogen activator inhibitor 1 or plasminogen activator inhibitor 2, pi1 and pi2. Also, inhibition of plasmin, the plasmin that were being produced, okay, to degrade the fibrinogen can also be inhibited by what? Antiplasmin. Okay, there's also inhibition of binding of exposed lysine residues, okay, of TAFI or using lysine analog. So the binding of fibrin lysine residues of the fibrin molecules to the plasminogen can also be inhibited so that there is no stimulus to indicate fibrinolysis to go ahead. So the tissue TPA is synthesized in endothelial cells. It's also stored in small dense granules in the endothelia. TPA is rapidly mobilized in response to stimulus. So the coagulation system is a comprehensive mechanism of forward and backward reactions, control mechanism. As the coagulation mechanism sets on, there are regulations to stop the clotting at a certain limit. And there are also regulations to stop the lysis of the clot that is formed at a certain limit. So it's a highly complex control mechanism. And some products of it are used in the evaluation of some disease conditions. All right, thank you. So If you have any questions now, you can ask.